All right, so now we're going to take a look at these different classifications of bacteria. So first we're going to start with our Proteobacteria. And I think I want to point out here these learning objectives here are, I think, very helpful in this case. It's kind of breaking down the information that we're going to take a look at and can kind of help you as we move from section to section, kind of keep things organized. So what we're going to be taking a look at these unique features of each class within the phylum Proteobacteria. So kind of keeping things organized. We're talking about the domain bacteria, and then we're talking about the phylum Proteobacteria. And then we're going to talk about these classes, each class within the phylum Proteobacteria, which within the domain bacteria. Okay, so we'll talk about the AlphaProteobacteria, BetaProteobacteria, GammaProteobacteria, DeltaProteobacteria, and Epsilon Proteobacteria. So those are the different classes within the phylum Proteobacteria. And then also you should be able to give examples of bacterium in each of these different classes of Proteobacteria. Okay, and keep these learning objectives in mind as we go through the next several chapters, because it really kind of, I think, keeps things very organized. So you can kind of take a look at 4.2 is talking about phylum Proteobacteria. These are the different classes within that, and then you can look through the chapter, read through the paragraph, and figure out the different bacteria and how they fit into that type of Proteobacteria class and then phylum. All right, so in 1987, the American microbiologist Carl Woese, which we've already talked about, suggested that a large and diverse group of bacteria, which he called purple bacteria and their relatives, should be defined as a separate phylum within the domain bacteria. And so that's what we have now. So we have our domain bacteria, and one of the phyla in there is Proteobacteria, which is what it's called now. It includes many bacteria that are part of the normal human microbiota, as well as many pathogens. So then we're going to further divide this Proteobacteria into 5 classes. So the alpha, the beta, the gamma, the delta, and the epsilon Proteobacteria. All right, so let's take a closer look at the AlphaProteobacteria quickly here. The first class of Proteobacteria is the AlphaProteobacteria. So the unifying characteristic of this class is that they're oligotrophs. So these are organisms that are capable of living in low nutrient environments, such as deep oceanic sediments, glacial ice, or deep undersurface soil. So you should know that AlphaProteobacteria are oligotrophs, which are organisms that are capable of living in low nutrient environments. So among the AlphaProteobacteria are 2 taxa. Okay, so we're going to then further separate the AlphaProteobacteria, this class, down into taxa, which are the Chlamydias and the Ricketsias. Okay, so now we're breaking that down further. These are obligate intercellular pathogens, or intracellular pathogens rather, meaning that part of their life cycle must occur inside other cells, called host cells. So the Chlamydias and the Ricketsias have to have part of their life cycle inside of other cells. Obligate, meaning they have to. It's an obligation. It's what has to be done. And then intracellular, within the cells, pathogens. When not growing inside a host cell, Chlamydia and Rickettsia are metabolically inactive outside of the host cell, so they are not active. They cannot synthesize their own ATP, therefore they have to rely on cells for their energy needs. So since they cannot make their own ATP, they have to get inside of other cells and use up their energy, their ATP. The Rickettsia species include a number of serious human pathogens. So for example, Rickettsia causes Rocky Mountain spotted fever, which is a life-threatening form of meningoencephalitis, which is inflammation of the membranes of the brain. So the Rickettsia, the R. Rickettsii, infects ticks and then can be transmitted to humans via a bite from an infected tick. So you should know some different examples. So for example, the Rickettsia rickettsii causes the Rocky Mountain spotted fever, and it causes meningoencephalitis, and then that is because the Rickettsia rickettsii infects ticks, that then the ticks can bite the human, and then that comes from the tick. Then the human gets that disease. All right, another species of rickettsia, Proezekii, is spread by lice. So remember, the rickettsia is a very, very tiny prokaryote, so it's infecting the ticks and it's infecting the lice. And in this case, it's causing the epidemic typhus, which is the severe infectious disease common during warfare and mass migrations of people. So this guy infects human endothelium cells, causing inflammation of the inner lining of blood vessels, causes high fever, abdominal pain, and sometimes delirium. So it's a relative of R. typhi, causes a less severe disease known as murine or endemic typhus, which is still seen in some southwestern United States during warm season. So we still see these things. It's not like they're totally gone. We don't see them as often, however. Chlamydia is another taxon of the AlphaProteobacteria. So we just talked about the rickettsia, now we're talking about chlamydia. So members of this genus are extremely resilient to the cellular defenses. So they give the ability to spread from host to host rapidly via these elementary bodies, as they're called. So the metabolically and reproductively inactive elementary bodies are an endospore-like form of the intracellular bacteria. They enter the epithelial cell where they become active. So again, remember, these have to be within a cell. They don't make their own ATP. So you can actually see down here, and then we'll talk about the next one. This is the life cycle of chlamydia. So you can see here's the epithelial cell, just a happy little epithelial cell in the human body. And then we get these elementary bodies. These elementary bodies are metabolically inactive, but they can invade this epithelial cell. And then what they do is they form these reticulate bodies. These reticulate bodies then are going to grow and multiply and be within an inclusion inside of this epithelial cell. And then these reticulate bodies are going to produce more of these elementary bodies, and they're going to multiply, make lots of them, and then these elementary bodies are going to burst out of the cell and then infect the next epithelial cell. So they have to be within there, and this is the life cycle of the chlamydia. So remember, you have to have the life cycle within a cell. Otherwise, it's not able to live outside of the cell. It cannot live on its own. C. trachomatis is a human pathogen that causes trachoma, a disease of the eyes, often leading to blindness. So that's another one. Trachomatis also causes the sexually transmitted disease lipogranuloma venereum, so LGV. So this disease is often mildly symptomatic, manifesting as regional lymph node swelling, so some swelling of the lymph nodes because our immune system is trying to fight it off, or it can be asymptomatic, but it's extremely contagious. These elementary bodies get around very easily, and it's common on college campuses because they do move around where there are lots of people that are close by. All right, so then here we have a nice little table here, Table 4.2, that talks about some different types of AlphaProteobacteria. So you should be familiar with these. So here is our genus, our specific type. We have our Agrobacterium, and down, I'm not going to read these out loud, you can read them, it's pretty basic. We have our microscopic morphology, so what they actually look like. So remember, when we're talking about our proteobacteria, we're talking about gram-negative bacteria. So these are all going to be gram-negative, as you can see. And then it describes it further. So bacillus, remember that's rod-shaped. Coccobacillus, the spherical round shape, and on and on. So then we can see our unique characteristics of these different types of genus of the AlphaProteobacteria. You can see that the Agrobacterium is a plant pathogen, agro, like agriculture, so it's a plant pathogen. And then you can see it gives us an example of a specific species, the tumefaciens, causes tumors in plants. Okay, so you should know this information. All right, so then we're going to move on to our BetaProteobacteria. So moving on to BetaProteobacteria, our second one after AlphaProteobacteria. Unlike the AlphaProteobacteria, which survive on minimal amounts of nutrients, BetaProteobacteria are eutrophs or copiotrophs, which means that they require copious amount of organic nutrients. So again, unlike the alpha that we just went through, the alpha Proteobacteria here, that are oligotrophs, remember, so oligotrophs meaning that they can live in low nutrient environments. Now when we're talking about the beta proteobacteria, it's the opposite of that. We're talking about those that need lots and lots of organic nutrients in order to survive. So beta proteobacteria often grow between aerobic and anaerobic areas. So aerobic meaning has oxygen, and anaerobic, an meaning without an aerobic oxygen areas. So for example, in mammalian intestines. Some genera include species that are human pathogens, able to cause severe and sometimes life-threatening disease. So the genus Neisseria, for example, includes Neisseria gonorrhea, which is the causative agent for the STI gonorrhea. And then we have also Neisseria meningitidis, which is the causative agent of bacterial meningitis. Bacterial meningitis. So when we're talking about proteobacteria, they need lots and lots of organic nutrients. And in lots of cases, that happens inside the human body, where we have lots and lots of organic nutrients. Like the example for Neisseria species, or genera, or genus, the N. gonorrhea, and then meningitidis. Neisseria are cocci that live on mucosal surfaces of the human body. So that's why the two examples that were just given for Neisseria were both things that are on mucosal surfaces in a human. They are fastidious, which is difficult to culture, and they require high levels of moisture, nutrient supplements, and carbon dioxide. So these are very needy bacteria, proteobacteria. Also, Neisseria are microaerophilic, meaning that they require low levels of oxygen. So micro meaning small, and then aero for oxygen, and then philic meaning loving. So they love small amounts of oxygen. So they require low levels of oxygen. For optimal growth and for purposes of identification, Neisseria species are grown on chocolate agar. However, when you think of chocolate agar, it's not actually chocolate that we're talking about. It's actually agar that's supplemented by partially hemolyzed red blood cells. So it looks like chocolate because it has burst red blood cells in it. So not to be confused with actual chocolate. Chocolate agar has these partially broken open red blood cells. So it gives it kind of this brownish tinge to it. Their characteristic pattern of growth in culture is diplococcal. So remember, diplococcal, di meaning two, and then cockle, which is the sphere shape. So pairs of cells that are resembling coffee beans. So we can scroll down here, we can see right here we have some Neisseria meningitidis growing in colonies on a chocolate agar plate. So you can see it looks kind of brownish. This looks a little bit more orange, but and then you can see the diplo here, where they have the two. They oftentimes grow in these kind of little pairs. So the pathogen responsible for pertussis, pertussis meaning whooping cough, is also a member of the beta proteobacteria. The bacterium Bordetella pertussis from the order Burkholderealis produces several toxins that paralyze the movement of cilia in the human respiratory tract. And it directly damages the cells of the respiratory tract and it causes that severe cough. So if you've heard of whooping cough or pertussis, it's where a person coughs and coughs and coughs so much that they can actually break ribs, for example. And that's because the toxins that the Bordetella pertussis produces actually paralyzes the cilia in our lungs. So we feel like we just keep needing to cough because anytime we cough, it's not bringing up anything. We don't have the cilia in our respiratory system that's helping to bring up stuff that we're trying to get out of our bodies. So we just keep doing that over and over again. So we have then this chart here, this table 4.3, that summarizes some of the beta proteobacteria. Here we can see, here's an example of the genus. We have Bordetella, which we just saw, Burkholderia, Neisseria is down here. So you should know these examples of the genus for the BetaProteobacteria. And then we have our morphology. So again, we're talking about gram-negative. You can see it's all gram-negative because we're talking about the Proteobacteria. And then you can see the differences here that we've talked about in previous chapters. And then you should know the unique characteristics of these. So, for example, Bordetella, the Bordetella pertussis causes pertussis, things like that. All right, so then we're going to scroll past here and we can move on to our third group, which is GammaProteobacteria. All right, so the most diverse class of gram-negative bacteria is the GammaProteobacteria. All right, so very, very diverse. So remember, we have our alpha, our beta, and our gamma proteobacteria. It includes a number of human pathogens. So for example, a large and diverse family, Pseudomonas, or Pseudomonaceae, includes the genus Pseudomonas. Within this genus is the species P. aeruginosa, which is Pseudomonas aeruginosa, which is the pathogen responsible for various infections in various regions of the body. So P. aeruginosa is strictly aerobic, so again, it needs oxygen in order to survive. And then it is non-fermenting, and it is highly motile. So motile meaning moves. It moves a lot. It often infects wounds and burns and can be the cause of chronic urinary tract infections. Also can be an important cause of respiratory infections in patients with cystic fibrosis or patients on mechanical ventilators. So we see this, it's kind of a very general thing. Wounds and burns, urinary tract infections, respiratory infections in people that have cystic fibrosis or patients with mechanical ventilators. So it's aerobic and it moves around a lot. So we see it in a lot of different places, this Pseudomonas aeruginosa. Infections by P. aeruginosa are often difficult to treat because that bacterium is resistant to many antibiotics. So we see these in these areas, and that's difficult to treat because it's already become resistant to these things. And it has a remarkable ability to form biofilms. So remember, biofilms are one way that bacteria can protect themselves by creating these thick groupings and creating this film that makes it very difficult to kill every single one of them. Other representatives of Pseudomonas include the fluorescent bacterium, P. fluorescens, and the soil bacteria, P. putida, which is known for its ability to degrade xenobiotics, which are substances that are not naturally produced or found in living organisms. So here, the Pasturellacea also includes several clinically relevant genera and species. So again, we're talking about GammaProteobacteria, and we're giving examples of that. So we have our Pseudomonas genus, and then now we're talking about the Pasturellacea genus. And this family includes several bacteria that are human and or animal pathogens. So for example, we have Pasteurella hemolytica, which causes severe pneumonia in sheep and goats. We have our P. multocida. It's a species that can be transmitted from animals to humans in bites, causing infections in the skin and deeper tissues. Then we also have our genus Haemophilus, so Pasturellacea, up here Pseudomonacea, and now we're talking about Haemophilus. It contains two human pathogens, so H. fluenza and H. ducreyi. So H. fluenza does not actually cause influenza, which is a viral disease which causes flu. H. influenza can cause both upper and lower respiratory tract infections, so including sinusitis, bronchitis, ear infections, and pneumonia. So when we're talking about Haemophilus influenza, the bacterium, the Proteobacteria, negative gram staining, we're talking about something that gives respiratory tract infections in the upper and lower respiratory tract. When we talk about like the typical wintertime flu, that is a viral disease. And we'll talk about viruses in a couple chapters. So before the development of an effective vaccination, strains of H. influenza were a leading cause of more invasive diseases like meningitis in children. So it was a little bit more severe. H. ducreyi causes the STI known as cancroid. So the order Vibrionalis includes the human pathogen Vibrio cholera. This is a comma-shaped aquatic bacterium. And so again, we're talking about, now we're moving on to the order of Vibrionalis, talking about more GammaProteobacteria. Remember, the GammaProteobacteria is very diverse up here. So we're looking at all these different types of things, the Pseudomonacea, Pasteuracea, the Haemophilus, now the Vibrionalis. It is, again, comma-shaped aquatic bacterium that thrives in alkaline environments. So now we're talking about an alkaline environment like shallow lagoons and seaports. And so this is interesting. A toxin that's produced by V. cholera causes hypersecretion of electrolytes and water in the large intestine, leading to profuse watery diarrhea and dehydration. So it's actually the toxin that's produced by that bacteria is going to cause this hypersecretion, increased secretion of electrolytes and water in the large intestine. And that is what is actually leading to the diarrhea and the dehydration. Here, our Parahemolyticus is also a cause of gastrointestinal disease in humans, whereas E. vulnificus causes serious and potential life-threatening cellulitis, which is an infection of the skin and deeper tissues. It's an inflammation of cellulite and bloodborne infections. So another representative of Vibrionalis, Lea Vibrio fishery, engages in a symbiotic relationship with a squid. So this is what is illustrated down here. The Lea Vibrio fishery is a bioluminescent bacterium, which is interesting. The squid provides nutrients for the bacteria. So here's our squid here in B here. Our squid is providing nutrients to the bacteria, and the bacteria are able to grow using those nutrients. And then the bacteria produce bioluminescence that then protects the squid from predators. So it can make it look shiny and protect itself from predators. So again, these GammaProteobacteria, we're going through these so that you have different examples of these proteobacteria that you're responsible for knowing, but then also the specific examples that are given here of the GammaProteobacteria are to show, for example, like Pseudomonas is, like a chronic urinary tract infection bacterium, versus Haemophilus, which gives us influenza and also bronchitis, ear infections. And then when we come down here in the same group, in the GammaProteobacteria, we have something like Lea Vibrio, which is Lea Vibrio fishery, which gives bioluminescence in a squid in the sea. So very, very different types of bacteria in this group. So moving on to the next one, we have Legionella, which is our next genus of the GammaProteobacteria group. So L. pneumophila, the pathogen responsible for Legionnaire's disease, is an aquatic bacterium that tends to inhabit pools of warm water, such as those found in the tanks of air conditioning units in large buildings. So this is B here. They're showing an air conditioning unit on top of a large building. And then here in A is our Legionnaire's disease bacteria, so our L. pneumophila. And because the bacteria can spread in aerosols, outbreaks of Legionnaire's disease often afflict residents of a building in which the water has become contaminated with Legionella. So these bacteria derive their names from the first known outbreak of Legionnaire's disease, which occurred in a hotel hosting the American Legion Veterans Association convention. And then that's where that came from, because in that case, in Philadelphia, this hotel had the water was contaminated with Legionella, the Legionella pneumophila, and then it caused that Legionnaire's disease. All right. Next, we have Enterobacteriaceae. This is a large family of enteric, meaning intestinal bacteria. In the GammaProteobacteria group, they are facultative anaerobes and are able to ferment carbohydrates. Facultative anaerobes meaning using oxygen to varying degrees depending on what's available. Within this family, microbiologists recognize two distinct categories. So the first one are the coliforms, One example, Escherichia coli. So coliforms are able to ferment lactose completely. So for example, with the production of acid and gas. So they use lactose as their food, as their energy source. The second category are the non-coliforms. So non-coliforms, which cannot ferment lactose, or can only ferment it incompletely. So producing either acid or gas, but not both acid and gas. The non-coliforms include some human pathogens such as Salmonella species, Shigella species, and Yersinia pestis. So E. coli has been perhaps the most studied bacterium. We hear a lot about E. coli. Since it was first described in 1886 by Theodor Escherichia, many strains of E. coli are in mutualistic relationships with humans. So oftentimes, perhaps when we think of E. coli, we think of the one that makes people sick, or the ones that make people sick. However, most of them are in a mutualistic relationship. Remember, mutualistic relationship is when both benefit. However, some strains do produce the potentially deadly toxin called Shiga toxin, which perforates cellular membranes in the large intestine, causing bloody diarrhea and peritonitis, which is inflammation of the inner linings of the abdominal cavity. So those are the ones that we talk about that can be very serious. So some E. coli strains produce a Shiga toxin, which can cause the bloody diarrhea. It's perforating those cellular membranes, so it's bursting cells. And then other E. coli strains may cause traveler's diarrhea, which is less severe but more widely spread disease. That's the one that I think most people are most familiar with, is getting an E. coli infection. Then you get some diarrhea as your body's working through getting rid of that, and it's usually a little bit more short-lived. The genus Salmonella, which belongs to the non-coliform group, so remember E. coli, kind of easy to remember, coli is coliform. Salmonella, now we're talking about a non-coliform group of Enterobacteriaceae, is interesting in that there's still no consensus about how many species include. So in the Salmonella group, we do know a lot about several Salmonella species. However, they're not entirely sure. They're still trying to categorize them, basically. So scientists have reclassified many of the groups that they once thought to be species as serotypes, also called serovars. So these are strains or variations of the same species of bacteria. So they're different types of the same bacteria. Their classification is based on patterns of reactivity by animal antisera against molecules on the surface of the bacterial cells. Okay, so when they are taking a genus, then they're taking a certain type, like Salmonella. When they take a species of Salmonella, thought to be species as serotypes. They once thought that they were species, like individual species. Now they're looking at serotypes because they react to different animal antisera. So serotypes or serovars are different strains or different variations of the same species. And then they react to different types of animal antisera based on the molecules that are on the surface of their bacterial cells. So a number of serotypes of Salmonella can cause salmonellosis, characterized by inflammation of the small and large intestine. It gives you fever, vomiting, and diarrhea. We know Salmonella poisoning. The species S. Enterobacterica, which is serovar typhi, causes typhoid fever, so with symptoms including fever, abdominal pain, and skin rashes. So typhoid fever is actually from a serovar, which is the typhi serovar, which is actually Salmonella species. All right, and this is actually a nice image here of the Salmonella typhi, which is the causative agent of typhoid fever. And then we're going to see here we have another one of these nice tables that is summarizing some of the GammaProteobacteria, some other ones that we hadn't already talked about. You should be familiar with all of the ones that we just talked about and have that information. And then here are some other ones here. although we did talk about Enterobacter. But here are some more GammaProteobacteria. We did talk about these. So Escherichia is there for the E. coli, the coliform, Haemophilus. We didn't talk about Klebsiella yet. Legionella. Proteus, we talked about Pseudomonas. Down here, Shigella, Vibrio. And then we didn't really talk about Yersinia pestis, but also in there. So, all right. Now we have Delta Proteobacteria. So again, we're moving through these Proteobacteria groups, the 5 groups. We did Alpha, Beta, Gamma, now we're doing Delta Proteobacteria. This is a smaller class of gram-negative Proteobacteria that includes the sulfate-reducing bacteria, or SRBs. They are named that way because they use sulfate as the final electron acceptor and the electron transport chain. So if you recall back to your studying of the electron transport chain, where electrons are passed from one thing to the next thing to the next thing to the next thing, and we produce ATP through that process. The final electron acceptor for the Delta Proteobacteria is sulfate. And so they are sulfate-reducing bacteria. Very few of the SRBs are pathogenic. However, SRB Vibrio oral is associated with periodontal disease, so disease of the gums. Delta Proteobacteria also includes the genus Vibrio, which is a species of which are parasites of other gram-negative bacteria. So Vibrio is going to invade the cells of another bacterium and then position itself in the periplasm. Remember the periplasmic space, right? So the space between the plasma membrane and the cell wall, and then it's going to hang out in that periplasmic space in the periplasm, feeding on the host proteins and polysaccharides. And then this infection is lethal for the host cells. So Vibrio is small, and it actually infects and invades other bacterium, getting into that space between their plasma membrane and the cell wall, and then eating up proteins and polysaccharides. Another type of Delta Proteobacteria, Myxobacteria, lives in the soil. It's a scavenging inorganic compound. So it's going to be looking to metabolize inorganic compounds while it's living in the soil. It is motile, so it does move around, and it's highly social. So they interact with other bacteria within and outside of their own group. Here you can see they can form multicellular macroscopic, meaning we can see them with our eyes without having to use any microscope. These macroscopic fruiting bodies. And you can see in the image down here, this whole thing here is a fruiting body made by this myxobacteria. So these fruiting bodies are structures that are still being studied by biologists and by bacterial ecologists. These bacteria can also form metabolically inactive myxospores, so similar to endospores where they're metabolically inactive, and then they're hanging out here with the active fruiting body. So here, remember the Delta Proteobacteria is a small group. We have the different types here that we talked about, and you should be familiar with those. And then we're going to move on to our Epsilon Proteobacteria. So the smallest class of Proteobacteria is the Epsilon Proteobacteria. They, again, are gram-negative, microaerophilic. So remember, micro meaning small, aero meaning oxygen, and philic meaning loving. So they like a small amount of oxygen or low oxygen levels. They remind you of that here. They only require small amounts of oxygen in their environment. There are two clinically relevant genera of these. We have our Campylobacter and Helicobacter, both of which include human pathogens. So Campylobacter can cause food poisoning that manifests as severe enteritis, which is inflammation of the small intestine. This condition, caused by the species C. jejuni, or jejuni, Campylobacter jejuni, is rather common in developed countries. So we do see it, usually because of eating contaminated poultry products. So we see a lot of Campylobacter jejuni in poultry products. And this is because chickens often have this bacteria in their gastrointestinal tract and feces. And then when people are processing poultry, processing chickens, their meat can then become contaminated during the processing. So during the processing of getting the muscle, we can get some of that gastrointestinal tract stuff in there, and then that can cause an infection and causes that severe enteritis, food poisoning. Within the genus Helicobacter, we have our helical, as the name implies, flagellated bacterium. So H. pylori is a very common and well-known one. Identified as a beneficial member of the stomach microbiota, but is also the most common cause of chronic gastritis, so inflammation of the stomach there, causing gastrointestinal pain, and ulcers of the stomach and the duodenum. So this Helicobacter pylori is what we've seen as responsible for ulcers. Studies have also shown that H. pylori is linked to stomach cancer. So we've seen that it's actually beneficial and it's helpful in our stomach. However, when things change, when environmental conditions change, then we can see that it can actually cause some stomach upset. It can cause ulcers in a long-term situation in the duodenum in the stomach, and then long-term it can cause stomach cancer. So Helicobacter pylori is actually somewhat unusual because it can survive in this highly acidic environment of the stomach. So it's a little bit I'm forgetting the word right now, but it can live in extreme environments. An extremophile, that was the word I was looking for. It's a bit of an extremophile because it can live in this highly acidic environment. It produces urease and other enzymes that modify its environment to make it less acidic. So it can live in this acidic environment, and then it just produces these things to make the area around it a little bit less acidic. And here is a picture here of Helicobacter pylori, which can cause that chronic gastritis, leading to ulcers and stomach cancer. Here's our chart for the Epsilon Proteobacteria here. So our Campylobacter and their Helicobacter. You should have this information and know that information. We just talked about it. All right, so next we're going to move on to our non-Proteobacteria, gram-negative bacteria. And then we'll also talk about the phototrophic bacteria. So we just finished up the section on Proteobacteria that is a gram-negative bacteria.